Hi, I'm John Slattery. I'm the Vice Dean for Research and Graduate Education in the School of Medicine. It's a pleasure on behalf of the school and the Council on Research and Graduate Education to welcome you to this Science and Medicine lecture. Really, it's a pleasure to introduce Alex Greninger today. I always get to learn new things about people. Alex, number one, you have entirely too many degrees. I'll go through that list. We crossed paths. Well, we didn't cross paths in time, but geographically, in that we both have associations with Austin, Texas. The list of degrees starts with a BA in International Relations from Stanford, and then a year later, Master's in Philosophy and Epidemiology from Churchill College, Cambridge. Did I leave something out? You also had a BS in Biology, I believe, from Stanford. I did leave something out. Then a Ph.D. in Biomedical Sciences from UCSF and MD then following that at UCSF as well. He continued UCSF to do a postdoc in Charles Liu's lab and also a residency at University of Washington. He then stayed here, joining the faculty in what's now the Department of Laboratory Medicine and Pathology in 2018. He's the assistant director of the virology division in that department. He's had quite a career so far. Last year, I'd like you to know among his honors is that he and Keith Jerome were co-recipients of the School of Medicine's Inventor of the Year Award for the very important intervention that they put, or tool that they put in the hands of everybody, but particularly those of us here in the Pacific Northwest, in being able to diagnose COVID infections with an mRNA test very early on. He's received a lot of other recognition. In 2020, the Pathologist Power List Award. 2017 40 under 40 for the American Society of Clinical Pathology, and one that I really like, 2004 a Winston Churchill Scholarship. And of course, as Winston Churchill was a student, he was not known for his scholarship. So I particularly like that award. He's also only published 180 or so research publications so far. And I think looking at that list of publications, it's accurate to say that Alex has a broad interest in anything that improves diagnostics and enables us to treat or prevent viral infections. And the title of his talk, as you saw, I guess I changed the slide, is That Virus? And I guess we can get which virus you'll be talking about. Thanks, Alex.
Thanks. Thank you so much, John, for that kind introduction. I'm it's a great honor to be able to give this talk, and I'd like to thank people who came here for Hognis. As John said, the talk is called That Virus, a little bit on SARS-CoV-2, which is, I think, everyone that virus right now, but also from the standpoint of the kind of work that we do, it really is broad in virology, so it can move around a lot. I'm going to move around a lot relatively quickly, trying to tell a few stories of what we've done over the last, since I've been here at UW, as well as a little bit of prior work as well. So my work sort of revolves around this sort of three-ring circus that I feel like I actually exist somewhere between South Lake Union, 1616 East Lake, and Harborview at all times. I'm actually sometimes at each space, but always somewhere between them, between the clinical lab, the clinical trials lab over at Harborview, and then my own basic science research lab over at South Lake Union. I'll try and touch on each here in this talk today. So I'm going to give a little bit of an origin story to start out with. For how I got interested, what I got interested in science, the science and medicine talk, how we've integrated pathogen genomics throughout. It's been a major sort of thrust of my work for the last 10 to 15 years. And then a little bit more on the operations for SARS-CoV-2, which has been more of an operations story than maybe a science story. There's always been lots of science, but for us in laboratory medicine, it's really been an operations work. So I got my start in science, or got interested in science, mostly around the idea that there were all of these new viruses to be discovered that could influence human health. We're really good when you find a new virus. They have RNA-dependent RNA polymerases sometimes. They have other enzymes that basically you have a wide therapeutic window. for what you can make an antiviral for. You can make vaccines. We're pretty good at it, actually, really good. It showed SARS-CoV-2, we're really good at it. And so being able to find those new viruses is very helpful for moving the case, be able to prevent certain diseases. And I took this picture from 2007, which is me with about 800 baby food jars of baby stool, because that's actually one of the best milieux to look for new viruses, as well as this is sort of my first year in the lab. And it's one of the projects that fell to me that basically someone thought some RNA was coming in and being extracted. Instead, they got 800 raw primary samples. So I took my Thanksgiving weekend in 2007. But to me, this really personifies or illustrates what viral discovery fundamentally is, and sort of come back to that at the very end. But in those samples, there actually were two samples that were positive for a new picornavirus, the first virus that I ever found, which we called classy virus, and another group called Sally virus. This is a typical picornavirus genome organization. It was sufficiently new to be its own new genus, which is pretty unique still at the time. They said it was in two twins at the same time, and it was the closest virus it was to this virus that most people don't study, but is a virus that's been found in Japan 20 years prior, called Aichi virus. And so we went on to do other work in Sally virus and classy virus, and showed the protease cut stuff. Could never get it to replicate in culture. It actually, other people have gone on and showed it's one of the most common viruses to be found in sewage, and is associated with pediatric diarrhea. But this was just a really interesting or fun start to viral discovery. And we kept doing that. We found there were several other astroviruses in that cohort that we were like just off by a little bit on in terms of finding first. And actually, sort of the next slide is one of the another big portions of what I do is about sharing data as quickly as possible. And a lot of that's some of this origin story that while we were putting together the genome, and I had never done this before, we ended up submitting to GenBank on May 21st, 2009. And another group actually also in San Francisco with Eric Delwart, a good friend, submitted the virus literally less than 24 hours before to the same database. And this was back when everything was totally censored. So you had no idea that these had been submitted at the same time. There's no way to know. You couldn't see it in the database. They would often go up a year later. And because this virus got named after their name, called Sally virus rather than classy virus. And so I've always got this very try to share genomic data as quick as, usually the ethic, but also is sort of from this origin story of trying to put things in there as quickly as possible. And we went on to do other work on the virus, as I said, and other biochemical work. But when I went back, that was my sort of PhD. And when I came back to medical school, I got hooked into clinical pathology and clinical microbiology. And there, rather than having to screen or deep sequence all of these samples to find a new virus, well, viral discovery is kind of a funny thing. The harder you work, the less impact you have. If it's not in the first 100 samples and you have to go down to 1,000, then your point prevalence is 0.1%. If you sequence 10,000, it's 0.01%. So the harder you work, actually, the less impact you're having, which is kind of a tough place to be. But in the clinical microbiology lab, there were all of these organisms, and I took one picture from UCSF of all of the bacteria that they had frozen down of very interesting genera and species that had, you go into, from a genomicist standpoint, you go in and look, and many of them had never even been sequenced before. It was back in 2013, 2014, where there was only one genome. And so I really sort of put a lot of effort into that year, basically digitizing these stocks, which I still get emails on from GenBank for people who find certain enzymes or they want, this particular isolate or whatever, and relate back to UCSF. And we found a number of stories over the years where this has been important. So one of the things that we were doing at that time was metagenomics, which is taking a sample, clinical metagenomics, where you take a sample and you shotgun sequence it and you align to everything that's in the database and you try to make clinical diagnostics on it. It's sort of like a broad range PCR, but it's sort of the broadest range that you can do. But at the time, and still during most of the 2010s, the database was not complete. So there weren't first genomes, there weren't second genomes. The genomes were sufficiently divergent. It was hard to align to them. And so filling out the database became really sort of something we really took on as a sort of almost a crusade, but a lot of work that we did put into this. And here's an example, actually. So we were sequencing the first genome of this amoeba, Balamuthia mandrillaris, which is a brain-eating amoeba that also eats other brain-eating amoeba. Ironically, it also eats Naegleria. And this was at the same time we were sequencing this particular organism and finishing out the genome for it. We got a case from Colorado Children's of a suspected unknown meningitis encephalitis case that we shotgun sequenced, and we're trying to figure out what it was. And it turned out to be a case of Balamuthia. But while at the same time we were sequencing the genomes, we were actually able to do this direct comparison where we were the only ones who had the genome because we hadn't finished it. But we could look at what was in the database. And what was in the database was just the 16S and 18S of Balamuthia, just little portions, just maybe 2 or 3 kb. And so if you look at we had CSF from the first day of admission as well as the 6th day of admission, and it was very easy to tell it was Balamuthia, whether you looked at the genome or 16S or 18S if you got the 6th day, but it was only in the context of the first day based on just, this is almost dumb, I don't say dumb luck, but just the way it worked out is that we put that many reads and made that library this way that you could find it only with that genome in the database if you added it in there. And so it's a very simple point, and this still goes to the point of where SARS-CoV-2 or other viruses or the next pandemic, the more that we find and we get into GenBank, the easier it is to be able to align to it as we sort of as more and more diagnostics actually do transition to this. So everyone only knows it's like shining the light in the lamppost. Here's another slide on this one, but basically, the bigger and bigger, we always hear about genomics and how there's more and more sequencing being done and more ability to sequence. But part of that actually is an element over here of discovery, where you're always looking a little bit broader, doing more distant alignments, assembling genomes, and then putting them in the database that now allows you to do faster and faster searches as you directly align against those things in the database, which adds to the utility of it. So part of making clinical metagenomics and part of the genomics revolution is getting these genomes in the database, in addition to all the science they affect. So actually, when I first came to Seattle, clinical pathology is it's a 3-year residency because ACGME says it has to be, but functionally it's about 15 months of required rotations. And so for the first 12 months there, I basically was continuing. So one of the things we knew is that there are a lot of new viruses to be found in animals and wildlife, and especially in insects. And so as I got to know Seattle, coming here, I made friends at the zoo. I made friends. One of the first samples we ever did was some geese from South Lake Union. We took some stool from them, shotgun sequenced it, found some new dicistraviruses. I made friends with the moth and the arthropod people, and we found a bunch of viruses and moths from here, as well as a spider over at the Burke. So a lot of spiders. We found a lot of new viruses. So I had actually had to reschedule opioid sign up to go to the zoo. People really questioned whether I wanted to be a clinical laboratory director during this time. It was kind of a very funny time. It was also a great way to get to know Seattle. So when you find these things, you don't actually know exactly what the host is, and you can name them whatever you want to. Ideally, you'd name them something related to the host and something to the virus. You're not really supposed to name it after people. But as I was here, I like sports. And so I actually started naming these viruses after people in Seattle sports figures. So Ken Griffey Jr. has a virus. Christian Rolzon has a virus. Nicholas Odero has a virus. Jordan Morris has a virus. Edgar Martinez has a virus, as well as fellows, ID fellows at the time. Elizabeth Duke has one. And eventually these will probably be renamed to an actual name associated with a host and a virus. But for right now, actually 5 to 6 years later, they're still their own species, their own genus of virus in a way, up there in the database out there. So anyway, I'm probably gonna letter for this, not putting this on YouTube, but nonetheless, this is what I did to sort of keep interested in viral discovery that we came here. Now, of course, we're in clinical pathology, and it's not just sequencing insects and arthropods and a lot of that actually came from bat stool. We got from paws, the animal rescue people. So you get bats and sequence them, and they're great aggregators, so they find all kinds of things. There were equal number of samples, just like at UCSF. There are all these different samples of frozen down harvest clinical culture isolates and specimens at the UW Clinical Virology Lab, which is really just second to none, just an incredible resource. And so we started digitizing those as well. And now looking back from the vantage point of 2021, this is actually what the viral database looks like. It's incredible. And more than 95% or 98% of viruses have been sequenced now are SARS-CoV-2. We're all about to hit 5 million viruses there that are isolates for SARS-CoV-2. Before that, it was just influenza, and everything else is almost nothing. This is sort of what that looks like when you look at from a database currently in 2021. It's absolutely remarkable. And I think I'm using this sort of to show we've always known there's been this disconnect between what we can do from a sequencing standpoint, and I'll get to the point of connecting it to all the things we get to use it for. But from a technological standpoint, we could easily sequence these things. It's really just people just haven't been interested in them. They have not done them. There's some of these, when we first got to HHV6, there were 2 sequences in the entire database for it. Just no one, this is a virus that's integrated into 0.4% of people's genomes worldwide. There's all kinds of fun biology, interesting things that people universally get exposed to it in the first few years of life. I mean, there's all kinds of interesting things that we can be done. And this was really amazing to me. And so we really focused on some of these viruses, the non-influenza respiratory viruses, as well as some of the herpes viruses early on, where we actually have done over half or sometimes 75% of all viruses sequenced worldwide have been done at UW Virology, just from our clinical virology lab, just sequencing those ones in the clinical virology lab. Actually, this is crazy. We looked at this going into the pandemic. Going into the pandemic, UW Virology was the leading worldwide sequencer of human coronaviruses, having done 58. Like, compared to SARS-CoV-2 and this explosion of genomic data, to be able to track it down to single mutations and look at variants and all this stuff, it's incredible what we don't know from these other viruses, just from a sheer number standpoint. Just if you just sort of like look at it from like stars in the sky and you just sort of zoom in and zoom out, it's incredible how little is done in these areas and how much growth there is given all the capacity and interest that's come out of SARS-CoV-2. It was sort of incipient in some of those things we knew we could do, we just hadn't done it that much. Now, sequencing really informs this, you can use it for many different aspects. I mean, right now in the SARS-CoV-2, it's been used for epidemiology. A lot, I just put a highlighted parainfluenza virus up here, but you can use it for epidemiology to look at, here are two sequences that are much closer, most closer time to most recent common ancestor than other ones. So they're probably more likely directly related if someone wants to confirm or refute transmission in a hospital ward or in a household. If you look at the therapeutic pipeline for this is just for parainfluenza virus. I'm using an example here. There's a couple of monoclonals in development, a couple of peptides and small molecule inhibitors for the attachment proteins, as well as other small molecules for the polymerases that we want to understand antiviral resistance to. We want to understand, what the barrier to that resistance is that would be helpful to bring in there. And then, of course, just from a protein structure standpoint, with all of the in silico modeling tools, having more sequences in the database helps you understand those structures even better. It helps you get towards function. I mean, you can always, people studying any question with this virus can use more data in the database when it comes to these genomes. So I'm going to share a few stories from just some of that early sequencing that we did for, so here's one on parainfluenza virus. Again, a virus that a lot of people haven't really appreciated. There are very few labs in the country that study human parainfluenza virus, but there's a ton of infections that we find. And we have no drugs, no antivirals really that are been approved for it. And we have no vaccines. So it's a huge gap. But we find tons and tons and tons of isolates in the clinical lab every year to parainfluenza virus 1, 2, 3, and sometimes 4. So one of the first things we did was we're just digitizing, just going along and sequencing, just sort of uploading and looking through these things. And one of the first things we actually found early on here's just a sort of a zoom in of a phylogenetic tree of some parainfluenza virus sequences. And you can see in blue, we've got the samples that we're sequencing directly from people. Now in green, we've got ones that are culture isolates. And the culture isolates are a little bit different because a lot of these guys are from 2009 or 2010, and I can't go back. We don't store samples that long, so I'm only sequencing things here in 2015 and 2016. But the first thing you can see is that we're seeing these low-level mutations in similar positions, only in the attachment proteins in all of the green guys, and almost never in the blue ones. So we're seeing this low-level mutation every time we're isolating in culture. And what's really interesting about this is that these are actually zero passage isolates. This is before you even, as you're isolating the virus, it is basically laying down these mutations that you don't see in clinical isolates. So we formally did this. We took a sample, or a few samples, a handful of samples here in blue, where we sequenced it directly from patients. And then we looked at when we isolated in culture, and you could see those same mutations come out all across the attachment protein. And some of these are actually surprisingly high level. And again, these are zero. This is not like passaging the virus in culture to see what happens to it over 2 or 3 or 4 weeks. This is literally as you get the thing growing. Like there's no way, almost inextricably, you can't pull it out. And as we map this to crystal structures, we can find certain mutations that have actually been found before. This histidine 552. We also have an entirely other site over here that's never been mapped in this virus that is clearly important in attachment or in fusion. Because we start putting these mutations in the context of biochemical assays, we're basically watching this virus increase it's being selected for increased fusogenicity. And this is a crazy experiment. I still find this one crazy to this day. But basically, here are some of the sort of negative controls. And this is right there, the wild type background. It's 1x, right? This is 1x of fusion going into cells. And you can see each one of these mutations increases somewhere between 20 to 100-fold as the selective pressure of culture radically changes what these viruses, have to do from a human standpoint versus what they're doing in culture. And we're mapping to their sites with these. It's just really, really a very intriguing example of all the pressures that we put in the clinical virology lab in culture. If we want to study these viruses, these are basically baked into the isolates that you're trying to study when you're making antiviral resistance. And I think what was really crazy was there were actually 2 additional patients where we found these mutations in this clinical isolate. So we went back to those patients and we're like, okay, well, we've only seen these in culture. We made sure it wasn't any sample swaps or anything like that, sequenced them many times. And it turned out that these were in our long-term immunocompromised patients that we get, where we really focus our respiratory virus diagnostics on over in the clinical virology lab. So these are patients who are getting stem cell transplants and solid organ transplants. And so you can see this individual here on the left is infected for about 300 days. So basically almost an entire calendar year that this individual is infected with pretty strong viral loads. Here we're showing the viral load on the y-axis. These are actually pretty strong viral loads throughout. And this was sort of a really backwards way to figure this out because we were looking at it from the context of genotype first. We're sequencing. We found this mutation we'd only seen in culture. We had never seen an individual. It turned out that this individual was part of a clinical trial. So again, this is how we figured out. We weren't doing this ex ante. We were doing sort of ex post. This individual was part of a clinical trial for a new neuraminidase drug. So it's a drug that basically cleaves off sialic acid. You can inhale it. It would cleave off sialic acid. And since most respiratory viruses use sialic acid for entry, it's sort of potentially a very interesting broad indication. You could use it for flu. You could use it for parainfluenza virus. You could use it for other respiratory viruses. And it sort of hadn't done great in influenza trials, but they were trying it in parainfluenza, sort of always on the cusp of working. And here's an individual where it got the drug twice. And this is right when it basically selected it twice for this mutation that we had never seen before in a patient. So, and we put this with our collaborators in Columbia and Moscona and Matteo Perotto put this into their sort of biochemical Olympics for their attachment protein experts for parainfluenza virus 3. And here we have, this is the drug basically cleaving off the sialic acids, preventing the attachment of the drug. And when we introduce these mutations into sort of an isotype, the actually patient-controlled samples and look at what's happening, this is a way to look at how well the virus is binding these sialic acids by looking at how well it's binding to red blood cells. And so here's a wild-type virus that basically binds to red blood cells, but it releases 60% of them really within the first minute of the assay. So it binds and releases, but there's a lot of releasing going on. If you just take that first mutation and you put it in, you can see it basically releases almost none of them in the first 0 minutes or 5 minutes or even 10 minutes. And some of it's holding on for 60 minutes. And it's similar for other mutations that we found throughout here. So it basically is, it's a very interesting, sort of for basic scientists, it's interesting way of the virus evolving around a host-directed therapy, which has long been one of the thoughts of host-directed therapies is they'd be very hard for a virus to evolve around, right? Because they have to have that sialic. Well, you never really get rid of 100% of whatever it is you're trying to inhibit, and the virus is able to change its kinetics to bind the remaining sialic acids that much better and adapt to that milieu, which is very, very, very interesting. And to do that just with excess clinical samples as we go along and are doing this sort of digitizing and then doing it in the clinical lab. So we have that close association with our clinical data as we go along. But this actually, we did this before actually any sort of sequencing studies have been done by the trial, which are still going on right now. Through this work, we've also sort of advertised our skills to other labs around the country, so more basic science virology labs. And here's another a collaboration where we actually got to work on it wasn't called molnupiravir at the time. It was called the Emory Drug Discovery Institute 2801 drug in influenza A virus in ferrets. And so this is now molnupiravir, which I think just got authorized yesterday in the UK. I forget where it was for SARS-CoV-2. This is Merck's new orally bioavailable antiviral. And so here you can see the drug working quite well at blocking viral replication in these ferrets. Here's the negative control, so it's where we're not getting blocked. You can see the fever curves work in a dose-dependent fashion. So if you give it with 100 mg per kg at 12 hours or 24 hours, you get much lower fever curves. If you give a lower dose or nothing, you get a much higher fever curve. And we're the genomics people, so we come along and basically look at some of these selections and work that's been done in the ferrets as well as in culture, and are able to show that basically you're getting this massive increase in the number of transitions of CDU and G to A transitions as you go through passage number, as well as this is a very frustrating drug to work with from a, a genotypic standpoint, because it is an air catastrophe sort of inhibitor, which it basically lays down, it sort of makes these sort of changes, not really random, but they seem to be random, across the entire genome at a low level all throughout the virus. And so it basically causes this air catastrophe, and now the virus can't replicate. Actually, it's actually impossible to select for resistance to it still to this day, which is something very exciting for molnupiravir as a drug. Same mechanism in SARS-CoV-2 really could work against a lot of RNA viruses. But here we only see that as we go from DMSO to slightly higher doses to slightly higher doses to sort of, almost mutations across the entire genome only at a very, very low level. So it's very, very difficult. Took a long time to do that work, but basically showing the mechanism in these ferrets and in cell culture are trying to adapt it to cell culture. So they're very exciting. We're still working with Georgia State and Emory on their other orally bioavailable antivirals as well. I mentioned briefly that we had done some of the work on coronaviruses earlier in terms of sequencing, and so I want to pull in a few stories from there. This actually, I mean, I sort of make light of it here, but this is, the question of, what if you just shine a very bright light on coronaviruses? But this has always been one of my favorite stories or interesting stories from a scientific standpoint of things, the weird evolutionary pressures that we put on viruses in the context of the clinic, especially in the context of cancer care and the SCCA, where basically we had sort of done this story 3 years before Trump posed the question in that sort of memorable press conference. Which is we actually had a case of an individual who had AML and needed a stem cell transplant. And when they're going to do a stem cell transplant, they will check you for respiratory virus, because the first thing they can do is not ablate your marrow, not knock out your immune system while you have a respiratory virus. So they'll try to let you fight off the virus and then do it later. This individual was infected with an human coronavirus 229E. It's an alpha coronavirus. And the AML was sufficiently advanced, and coronaviruses, based on data here at Fred Hutch and UW, has shown to be less likely to progress to pneumonia and lower respiratory tract disease. They said, okay, we'll just watch it closely, and we're going to go ahead with the stem cell transplant. So they went ahead with the stem cell transplant, and they took 2 swabs right here. Okay, so before the transplant, and then they come in with the cyclophosphamide and the 12 grades of total body irradiation, and they have 2 additional samples after that. And basically, we can watch the genome of this guy as it goes along. Here's a little PCR over this region right here that's going to get deleted, and you can see right between the transplants, right as the transplant is being done, basically makes the largest, what before SARS-CoV-2 was the largest deletion ever in an RNA virus. Is about, I forget, like 600 bases, about knocked out an entire gene. It's actually an accessory gene. Never been shown in people or in vivo to be non-essential for replication. Here we're showing it's non-essential for replication in people, but in a dish you can knock it out as well. And these are the genes actually that make SARS-CoV-2 so bad, in addition to its spike protein. But SARS-becoviruses bring so many other innate immunity antagonizing genes in these accessory genes. And here you can see basically that we can knock out these genes from total body irradiation. We think it's from total body irradiation. It could technically be from sort of lack of immune pressure. There's a lot of things you can't entirely control for when you're doing this in people. But it was one of those really sort of fundamentally interesting stories from a science standpoint, personally for me, because you show these things in people, as well as these crazy evolutionary pressures that we put on viruses in the clinic. And I actually think that microbiology could use a little bit more work with our radiation oncologists. We're doing this all the time in the radiation oncology suite, and there's lots of people interested in the microbiome. You actually can do gene essentiality screens in the context of radiation oncology throughout. But this is sort of, again, things we see in the clinical lab that other people, you wouldn't think to do this. And I'm not saying it's radically changing therapy or anything like that, but it's just for very interesting studies when there's people are not studying these viruses to any great deal. I'd also say that, those viruses that we sequenced before, this is one of the reasons I do it, because I don't know what we're going to want to do it for. But SARS-CoV-2 uses, as it was an example, a great story of showing how you can use this data for other uses. So here's a paper from Trevor Bedford and eLife looking at, the questions are like, okay, what's going to happen with SARS-CoV-2 evolution right at the beginning of the pandemic? And there's no way to run well, now we have ways of running the clock forward a little bit. But one of the ways to run the clock forward is to look at other coronaviruses and see. So now we've got all these data in the database. We didn't know there was going to be a coronavirus pandemic 5 years ago when we were sequencing these things. but we make a substantial portion of the sequences using these analyses. Or gene synthesis. If people want to make a nucleocapsid protein against sarbecovirus and make sure it doesn't cross-react with the other beta coronaviruses. That data has been used. Wes Van Voorhees used that data a lot to help synthesize those proteins. This is a great study from Jesse Bloom's lab, looking at how human coronavirus 229E evolved over time. So they went back and synthesized one from 84, 92, 2001, 2008, and there's one of ours from 2016. These are all ours down there, and used it to sort of show that coronaviruses actually evolve around the immune response within these windows, at least. They didn't do any higher resolution than this, but within sort of like a 5 to 6 year window, and probably even faster, but really biochemically show that the receptor binding domain mutations that are laid down are the way that you do that. And you can't do this unless you have that prior data in the database. So again, I just like to highlight this because it's the data goes on and has many, many, many uses. So that's an argument for kind of some of the work that we've done I've invested a lot of time over the last like 10 years, and for Sheila here in the audience, they're doing the same thing with pallidum, sort of build up the first 1,000 genomes in a way. Let's get to that number, and then we can start using that as a sort of a dictionary, a library for more functional assays to be able to look at how we can make sure our vaccines are covering the world, make sure our therapies are going to cover these different strains that are out there, get some idea that's out there first. And that's sort of one of our big contributions in the science and medicine standpoint. So that's just sort of an illustration of the things we've done in the clinical lab from the science standpoint. I wanted to go back to sort of the three-ring circus and talk a little bit about the clinical virology work as well as the clinical trials lab work that we've done for SARS-CoV-2. And I think one of the points that I kind of wanted to make here is that COVID-19 and SARS-CoV-2 has shown the importance of science and medicine to really, how could we ever, name any other field where you could have something emerge, a new disease emerge, and in less than 1 year have authorized therapies and vaccines, diagnostic tests, and basically be in the ability to try to begin to control it. And so it really is an incredible story of science and medicine. But from the standpoint of people involved in right now, what we're doing with COVID-19, there actually wasn't a lot of science to be done. A lot of it was straight up operations. When the virus emerged, I sort of pulled this timeline from ASM. There was originally these cases of unknown pneumonia in December 31st, 2019, when the China alerted the World Health Organization. Within a week, basically identified SARS-CoV-2. The genome goes online on January 10th. When the vaccines and even the World Health Organization diagnostic assay were basically designed over a weekend from January 10th to January 13th. And the Moderna vaccine, the Pfizer vaccine, a lot of our diagnostic tests that we still use today, basically were made over that 72-hour period from a science standpoint. Now we went to the bag and went and all right, what's the best thing? What are the best constructs? We have our prefusion work that's been worked out in the glycoproteins to make the constructs for the vaccine, mRNA technology, put all that together and basically begin the clinical, the study of them and the getting them through the FDA and the trials. But for the most part, this was with the science time. In one argument, you could say this is sort of the science time of it. Obviously, all of it's science and but a lot of it just came down to operations. So for instance, for us, when it came time to making an assay, the major thing that we dealt with was administrative or operational. It really wasn't a science question of making a qPCR to detect an RNA virus. When we were trying to make this assay, there were thought to be only 14 positive cases all the way up until about February 20s in the United States, in the entire country. And the FDA basically asked places to have 25 to 50. They changed the rules as they went along, which is great, very important. But there were not enough samples to really validate these assays, and you couldn't get access to them. You couldn't get access to the samples, the first samples here in Washington State, because they'd already gone to the CDC, and we couldn't get access to the assays from the CDC even when they were authorized on February 4th. There are a number of cases like UC Davis of community transmission that the CDC denied testing for because of the epidemiological criteria. We always had the slow turnaround time we had some slow turnaround time within the first 2 weeks of the first case. So in the latter half of January at CDC, the turnaround time was basically headed over a week, and we were already getting complaints of samples that were being sent out. There just was not diagnostic testing. And I said we couldn't get the samples. They were all at CDC, which makes complete sense. But the standpoint, we just still, it was, that was the problem with operational. We tried everything we could to try to get samples, couldn't do it. Cultured virus you got in the middle of February, but you couldn't work with a BSL2. And even just like, even at UW standpoint, you had to get it through the BS. You had to get a BUA to be able to have it, be able to order it, to get it, which would take a few weeks. And then the committee would have to meet, sign off on growing it, to titer it, just to send it to the clinical lab. So that wasn't even an option until the first or second week of March. We looked at in February for trying to even work with a cultured virus. And that viral RNA wasn't available from distributors until really the first week of March. And it was really all of the struggles during this month and this month and a half to make diagnostic tests, to try to get these samples, to be ready to do all the analytical specificity work. By the time we had a positive on February 28th, the virus beat all the processes, making our way to a laboratory, and we were able to validate and do the science of validation in about 48 hours and have the assay ready to go on March 1st. But it was fundamentally about, it wasn't a science problem, it's trying to get samples. And we still haven't actually solved this problem for the next pandemic. There's a lot of things we could do, but we still can't use in silico controls and other things like they can't synthesize controls right now for to get things through the FDA for EUA processes. After we did go live, UW virology is an incredible curve. I still don't believe it. This is from the COVID testing projects, samples tested from March 1st to March 14th. Here's what we actually tested. And you can see this goes up to 7,000, this goes up to 1,500. So on different days across the country, we're doing somewhere between 20 to 50% of testing in the country, or the COVID testing numbers are wrong, finding some of the first hundreds of cases. And we were taking samples from all over the country. This happened. Incidentally, we also did, our genome sequencing pipeline was going at the same time. We were doing 20 to 30% of worldwide genomes over the month of March 2020 and into April. This testing curve really tells so many different stories. I'm going to use it to tell a few stories here in a couple of slides, but I always keep track of this. We're right now at 70.6 years of testing in 20 months. We're currently running one sample at 3 RVUs, so it's like an office visit every 8.5 seconds in that lab. And this has just been such an incredible amount of work and accomplishment for the lab and the people involved in the lab, as well as just really after 20 months, it's exhausting. It's very, very difficult to keep up this sort of level of focus and activity. But we're doing it, and we just passed 3.5 million samples tested. Like I said, 50,000 samples be a typical molecular testing year for all analytes in the clinical virology lab in prior years. You can almost set your watch to that in terms of the number of samples we get in. So this has just been an incredible increase. And I'll use it to tell the operation story in a second here. From an actual, like, what this means physically, we've doubled the size of the lab. We're up to like 30,000 square feet. We're going to have to move down to Renton here pretty soon. Have a lot of different instruments, whether cobosses, and MP96 extractors, hologic pantrafusions, Alinity M's. All of them require, of course, according to College of American Pathologists, you have to test every single platform against every other one. So like it just builds this incredible intricate web of cross testing, which is great actually for academic, sort of showing how well they work against each other. But it's been an incredible, nothing quite like this. I think another thing that I really want to highlight here is we focus a lot on the tests and the analytics, but it's really the pre-analytics is where 90% of the problems are in testing. And so in our prior world, we would get samples into our virology lab and we'd have these 3 spots to receive them in. And now this is our pre-analytical operation for receiving, accessioning, labeling, making sure things go to the right, dealing with any problems, any spills, anything that happens in those samples that are coming in every day. We're processing 2 to 3 times more daily samples than the rest of all of UW medicine, lab medicine combined. And we have an entirely new 60-person sort of operation for processing those things 24 hours a day, right? Virology before was basically like a 14-hour a day operation, 15-hour a day. Now we've been 24 hours a day now for basically the entire 20 months. And all of the people, like just the physicality of that operation, the supply chain, the receiving of, crates of things and making sure people come in and people are happy and fulfilled and, hiring. This operations has just taken an incredible army at each spot along the way. I just wanted to highlight a few people here with sort of this forum. Arun basically helped make the first quantitative PCR test. We were doing that in January and February. Keith moved basically to the lab for the first 2 months there and was just an incredible leader during those first 2 months of the pandemic, from sort of just like just an army operations standpoint. Greg basically has managed the entire lab the entire time. He's overseen all of our development work during this time too as well. Jared has done incredible, and Kyle Wing has done incredible work on validate these tests and keep the operations going. I remember, I mean, this is the sort of things that happen. Like say, like this is someone we don't ever work over a third shift over midnight. And for the first 2 to 3 months, he's one of our most experienced sort of medical laboratory scientists. And so now he's the third shift supervisor working from 11 p.m. to 7 a.m., for 2 to 3 months there to make sure that the people who are working over midnights actually know what's going on. Pavitra's taken on all of really led all the sequencing work that we've done. Tamara, an administrator, I mean, just the administrative aspect here of getting through contracts, getting through hires, getting through MTAs. Jennifer Greenwood came back to the lab. Patrick Matthias, our new clinical vice chair of clinical operations, basically made this dashboard. He actually programmed this dashboard himself, along with informatics colleagues, and basically just made this whole operation work along with Jeff, our chair, and Marcelo, our administrator. We brought in tons of new people who came in and volunteered during the first month here. We basically went exponential the first 2 to 3 weeks here before we had to transition to sort of actually triaging samples. We stopped having to run outpatient samples from farther away places because we didn't have the supply chain for it. And Hannah took on that supply chain, is now the operations head for clinical laboratory medicine. Caitlin Barron's a business analyst from a consultant from informatics, came in and basically made our pre-analytical operations. So how a sample comes in, how it's received, that there's how people know that it's received, through the entire process. While we were at this time when we first got these samples in June and July, this is when the drive-throughs went live, as well as Jay Inslee said that basically every nursing home had to test within the first every 2 weeks, and they had never tested before. And they started FedExing. We received like 100 FedEx boxes of 100 samples. Some of them are just like parafilmed around the top. They'd all be spilled together, and like they're just nonstop just issues with them. But we got it done. They gave us 2/3 of the state, and the other third of the state was supposed to go to another lab, and the other lab tapped out after 2 days of testing. So we got to take the entire state on and basically make that work. And these are the people who made it. Jessica Wagner came from a Research Sciences 4, came and helped manage basically just the receiving and opening of packages and making sure the supply chain was going. Trevor basically wired all of these rooms to make sure that we could actually have hookups for the instruments as they got built. All their informatics colleagues, Noah and Nick, just the physical wiring and getting the wireless ready. Basically, that has just been, he's like lived there at virology. Brett also is an operations director, and then Aaron from the clinical from Harborview, who was sort of helping manage the operations at Harborview, came in to help basically fix, get our Panther instruments and other instruments up and basically do the QC, because it was just too much for any one person to take on. So they're almost like a second manager. People from my lab, Amina Detia helped set up he's a grad student now, helped set up all of our automation equipment for sequencing pipelines. Basically, we can be able to do tens of thousands of samples, or thousands of samples a week. Again, the administrative, just constant contracts coming through, managing MTAs, just that's been incredible. Michelle was an undergrad in the lab who programmed our variant calling pipeline that was now used for most of the FDA submissions for Janssen and Novavax. And then a few other people, Angela Yu, who came in and basically has hired almost more than 300 people, which I feel like deserves its own like university award in and of itself, for just trying to actually, people have gone through the hiring process here, but how do you actually hire hundreds of CLT, clinical laboratory tech ones, to come in, process samples, get them trained, get them competized, follow all the rules, and then people go off and get other jobs. And there's always just this normal churn of just kind of having to hire and hire and hire, especially in this environment, to make sure that we are actually able to pull off these 10 to 12,000 samples a day. Then at this time in February and March, we ended up getting the trials for Janssen and Novavax, and as well as some other ones. And some of these trials, actually, you don't get to, it's OWS. So it's very much like they need to like enroll now. They need the data of the FDA. EU had to go up as fast as possible. And some of these trials started to like basically converge. So you go get these waves that like hit together and like crest at the same time. So we had to convert the clinical trials lab to 24 hours a day. And call in Abbott, and we had the Army involved actually in some of our operations to make sure that we could get all of the reagents that we needed to be able to perform the testing for these clinical trials. And then one of the things I think this is remarkable from a laboratory medicine perspective, while you're doing all of this and you're so focused on the instruments and the tubes and the pre-analytics, is starting July 1st, 2021, we got a big help from Seattle and King County, the Seattle City office, in terms of managing those collections for all those drive-throughs where all the samples came through. And now, starting July 2021, we're managing all the collections, our collections coming to UW from around the state. So that's a new, like, 100 people that have to basically be hired, trained, competized, and spread around around, Aurora or Soto or West Seattle or all the different sites that we run, and make sure that we're providing these 10 to 12,000 samples a day to the lab. Now Delta obviously is a big impact there, but just to be able to have these numbers go up as high as they do is really Jennifer Greenwood in an incredible new role doing this work to manage those collections. And it's not many departments or groups that, and I sometimes can't even begin to think about it. I'm up here to talk to you about virology and how viruses evolve, but we have to actually make sure that those tubes come in and are correctly collected legally according to FDA rules or Washington state competized rules, and make sure that people are there every day to do that. And our department has risen to that challenge now as well. And honestly, we don't really see too much of a decline. I mean, there's a little bit of decline recently, but it's still staying quite high. If you look at the national numbers, you're seeing what's happening in Europe, which is often kind of front run the US. As it gets colder here, we're still only at 70% vaccinated. The winter's going to be rough, anticipate it. And so we need to be able to continue to collect these samples to help in the public health response. What Pavitcher's done from a sequencing standpoint and integration to that has been absolutely incredible. So we've got basically done 31,000 samples here. This is just for surveillance. This is not counting all the trials that we've done for different trials work. But this has also been great to be able to feed this directly into the UW science machine. So Michael Gale and David Wiesler and Wes Van Voorhees, and be able to use this data to identify individuals who are infected with certain variants, what the response to that variant is. Look at how it acts in mice, and we basically we have every single variant of concern or variant of interest has made its way somehow to our lab. We were some of the first ones to send some of these to the CDC or even BEI or ATCC, so other could get them. That's been just an incredible way to take what started as a very bespoke, small, 2 or 3 people, all manual, to a really industrial operation from the viral sequencing world. It's just been incredible to watch. Here's all the basically variants you see very clearly in Delta, but now the question is, different varieties of Delta and splitting and what's the next strain going to be. And that's, we don't know. No one actually knows. And that's why we continue to do this, even when you're like, oh, it's 100%, Delta here, but you really don't know what's coming next. One of my favorite things also, I want to give a shout out to the press office also as well. UW does incredible. My favorite things to do if I go home and, watch TV or just open the paper is to watch Ashish Jha give a talk or talking about something on CNBC or CNN or something like that, and up pops some B-roll of our lab. They've done an incredible job getting images and video out from our lab. This is just some of it here of the people in our lab doing sequencing or serology for the trials, or setting up more Abbott tests for the quantitative PCRs. This is sort of the clinical trials. It's hard. I really wanted to hold time on this because it's hard to talk about this. When you think about SARS-CoV-2, what we need to get therapies, and we need to get vaccines and diagnostics out to the people, and we have to do it in a way that is consistent with regulatory processes. And this is where we probably do some of the most important work. It's so much easier to talk about testing. We're so much focused on that day to day from a clinical testing standpoint. But from a force multiplier standpoint, our involvement in all these different trials is probably some of the most important work that our group does. And yet it's the hardest to talk about, because it's pretty quiet. Sometimes I don't even know if I'm supposed to say, what trials, but at some point it comes out and it's all in the FDA filings. And you just receive barcoded samples with no identifiers. You're involved in some of the original trial design stuff like that, but we just have to do this. These trials go for 2 years, and the trials keep changing. People add boosters, and now all of a sudden there's 3 new serologies that need to come through, and it's 30,000 people and 4,000 people have dropped out or something like that. And okay, we're going to add this, and we're going to send 26,000 samples. And today, the data, the database that receives this data for this one data transfer can't take that many characters. And can you move it, slide it over this way? And it's all very official, done with all these signed agreements and MTAs from all over the world. For for all of these different trials. And so from a science and medicine standpoint, it doesn't, sometimes it doesn't feel like science. It doesn't feel like science, but it is the most important thing that we can get these things through. And so I'm so proud of our group here that has just continued to pump out these results in a incredibly accurate and validated and sort of traceable way that could handle an FDA inspection for all of these different trials. And we had a major victory just and have every little day there's something new, something, someone's filing a biological license application as opposed to their EUA. So they have a new 3,000 samples that need to get done now. Novavax just got authorized in Indonesia, I think yesterday. So that's their first authorization. They're probably going to go a lot more authorizations coming forward in the next few weeks. So expect to see. But that was a huge amount of work the whole time, especially in February and March, where that was basically consuming some for most people like 24 hours a day of just the time of just getting these samples through as they come through. The remdesivir trial, which actually was an incredible trial, run here at UW as well. Didn't do any testing for the first year, and then we did the testing later. We still have other phase 3s and antivirals coming through, even hydroxychloroquine with Ruan and Christine Johnston on the precursor prophylaxis, as well as we've had some work with other diagnostic tests, some really highly leveraged diagnostic tests from antigen testing. Those things have to get through the FDA. They have to have essential testing. And our goal is to make that as easy and as fluid and as sort of cost effective as possible to help get these things out there. And yet, again, from a science and medicine standpoint, we don't really get to talk about it as much as we like. We read about the results in the paper, just like everyone else. We have no idea what's happening a lot of the behind the scenes. But this is where we've done the viral loads, qualitative viral testing for all of Novavax. Every result in Novavax trial from a virology standpoint, serology standpoint came through this lab at Harborview, as well as we're doing sequencing for determining if Novavax is 93% effective against variants of concern in their trial that they've released results in May and June. That's all done through this lab as well. And the same with these other ones. So I always find a hard time like going through each one of those, but when every number that you see in those FDA filing, every number, but most of the numbers you see in those FDA filings as well as in the paper basically was done by this group over at Harborview that I'm just couldn't be more proud of. And it's just an incredible use. So I wanted to finish on one last slide. I started with viral discovery. It's the reason I got into science, and I'm very excited about. And so I'm very excited about this new grant that we just got with WashU St. Louis and Washington State as the point. I showed you there with 800 samples of poop looking for new viruses. Well, we know what new viruses can do to us now. And so we just got this grant to go through basically around 1 million wildlife animal specimens screened for coronaviruses, paramyxoviruses, and filoviruses. It's just a bigger boat than what we were doing 15 years ago. It's a huge genome recovery and data sharing project with viral characterization here at UW from the Wiesler, Gale, Van Voorhees, and Baker labs, as well as Sean Whelan's lab at WashU St. Louis, as well as the USAID projects. We're building capacity in 12 countries around the world. A lot of bats, maybe some geese. I'm just excited to have it sort of come full circle. This is really one of the few viral discovery grants out there to be able to get and be able to get started. It's a lot of work, a lot of administration to start out with, but we're 1 month in and we're making good headway here. And with that, I just wanted to really say, I think the overall theme here from what we're talking about is science realized in the clinical lab. I try to put most of our scientific work in the context of the clinical lab. That's how I keep it translational, as well as how we translate all of our laboratory discoveries into clinical practice. So I love our department and I love what we're able to do. It's very exciting to be fused with pathology now, but I really am so excited about what we're able to do. And I want to give, I called out a lot of people already and sort of told little stories, but I wanted to put some pictures up as well. Again, thanks to Keith and Pavitra and Greg, both Bob, who just retired, and Jeff and Patrick for the amazing leadership. And then as well as my own lab here at South Lake Union. Then moved on to grad school in Michelle at Boston, but Nicole, Quinn, John, Curtis, and Kate and Ben over at South Lake Union have just done incredible work as well through this pandemic. And with that, I'm happy to take any questions, and I really want to thank you so much for inviting me to give a talk. Oh, did I hit escape? Oh, question. Okay. All right. That. Please place any questions. No questions. Okay, please. Yep. Yeah, that's a great question. So the question is basically on the zero passage isolates, as you're isolating viruses seeing these mutations. Now, they're not random because they're really in the exact spots that we showed increased mutagenicity. And the overall hypothesis is that when you put these viruses, usually you're taking pretty high viral loads that they're culturing out. There's a lot of virus there. And so there's a lot of virus for not many cells. And so these viruses are adapted to go all in on entry and fusion. And it's like a race, right? So that the first one to get in that cell, then they're going to replicate, 10,000, 100,000 fold. And then they want to spread to the next cell. So actually some of these guys can cheat and actually go cell to cell fusion even faster. And so you're selecting for just in that first isolation, that isolation of the virus, you're looking at viruses that are much more entry driven than sometimes exit driven, honestly. That's sort of the overall hypothesis. Maybe I butchered some of the biophysics of it, but that's why we think we see that increase in fusogenicity. So you are seeing evolutionary forces the second you're always seeing evolutionary forces, right? You just can't escape them. Or even when you say there's none, there's something, right? But that's what our thought is. Yeah, it's hard because there's a little bit. The question is, can you estimate how many times they multiply before you get these mutations? I mean, you can. The trouble is that there's sort of like you can look at the copies of the virus, and you can look at how much actual virus you get from plaques or areas that come out. There's a loss, usually about 100 to 1,000 fold between the copy number and what you get out. And so it's a little bit hard to look at the efficiency because you already have that loss. If there was a perfect one-to-one relationship, you could possibly do it. So we could probably make some guesses, but fundamentally, I always look at that difference between the copy number and what you actually get to isolate as being sort of, if there were changes in efficiency. We actually just showed this for SARS-CoV-2. We looked at different variants and how efficiently they grew out. What's the relationship between their copy number and how much virus you get? Actually, Delta is more efficient at growing out, which is sort of consistent with its ability to sort of infect better in culture and in people. And so, there's that element, but that's probably the hardest part to do that. So thank you for questions. I appreciate it. No.
Okay. One more thing, you get a certificate. I actually see that degrees in here, so I think you've got the record for that ratio. Okay?
Oh.
But thank you very much.
Thank you so much. I really appreciate it.
Oh,
Oh, no, we're not doing that. All right, cool. All right. Thank you very much. Thank you so much for coming. I really appreciate it.